Obrigada, Nájila.
Thank you, Najla. We would like to invite on stage Leonardo Ribeiro Batista Silva for his poster presentation. He will present the poster, Efficient Large-Scale Point of Production of Affordable and Anti-CD19 CAR-T cells for immunotherapy. Good afternoon, everyone. First of all, I'd like to thank the organizing committee for the invitation and for giving us the opportunity to prepare the work we've developed at the National Cancer Institute. And today I'll be talking to you about one of the projects that we have developed over at the National Cancer Institute, which is the production or manufacturing of CAR-T cells at the site where patients will be eventually treated. So Najla has already helped me with some concepts, but not going back briefly, what is the CAR? The CAR acronym and antigen chimeric receptor. And that's so because it's an artificial receptor that is a chimera in which you have qualities or characteristics of the T cell receptor with the antibodies. So you have a part of, SC part, FV part, which is able to recognize the antigen, tumor antigen, and a domain, intracellular domain for the activation of these T cells. And how does T cells get produced? Basically, you get the cells from the patient, you modify them genetically so that they'll be able to express the chimeric receptor. You expand these cells in ex vivo fashion, and then you reintroduce those cells in the patient's body. Currently, there are 4 products that are commercially certified in Brazil. These are products based on CAR-T cells, anti-CD19, and BCMA, which is CAR. Well, since we're at a symposium held by Biomanguinhos, I'd just like to highlight that we have a technology transfer agreement with Carencross, and these CAR-T cells will be produced at Biomanguinhos through a partnership agreement with the National Cancer Institute. Now, drawing on this on the viral vector production platform for genetic modification of the CAR-T cells, they'll be produced at Biomanguinhos, and we'll be able to do this manufacturing also here in Rio de Janeiro. Well, today the process of manufacturing has these features. It is based all the way from the leukapheresis of the patient, the preservation of leukocytes, and then you send these cells, frozen cells, to the site, to the manufacturing center. These cells at the manufacturing center, they are activated, and you expand these cells and you modify them genetically using viral vectors. This thing is working by itself on its own, I think. So upon genetically modifying these cells, you expand them ex vivo, and these cells go through a quality control process, and they're taken back to the hospital where the patients will be treated, and the cells will be infused in the patient's body. That's how the manufacture of CAR-T cells takes place in Brazil. This is how it happens. Now, the goal of our work is to be able to align and to do the manufacturing of these CAR-T cells efficiently and large scale at the site where these patients will eventually be treated, which is the National Cancer Institute. Well, our strategy to do that is not to use viral vectors. We're using non-viral strategies, transposon, transphorases. We have two components in the system. We work currently on two components, plasmodium components, one of which codifies transposon, and the other one is transposase, and the other through a mechanism of copy and paste in the genome. We're able to insert these transgenic cells in the genome of the KT cells. So there is an advantage and a disadvantage. There are pros and cons when it comes to the use of viral vectors. For example, I'm pointing here the disadvantages and the utilization of viral vectors. You have a production which is very cumbersome, very troublesome, and high cost. And the whole plant, the production costs are high. The whole plant that was set up to produce vaccines at Biomanguinhos is based on viral vectors. It will be turned into a venue for the production of vectors for the transduction of KT cells. And we have some risks also of activation of oncogenesis when you do this, especially when it comes to viral components. And commercial products use this kind of system of viral vectors. So we began, first of all, to do some small-scale data in which we show that we're able to generate these cells, and these cells are efficient in the control of the tumor. And we're showing here that these cells, 19 BBZ, are the cells with KT. So we have here the animals have been inoculated with a tumor that's able to, a tumor cell that's capable of expressing luciferase. So we have the bioluminescence here following up on the tumor progression, and the animals that were treated with KT cells. You have here the regression and the progression of that tumor. When we look at the survival rate of these animals, we see that the animals treated with KT cells generated, even in small scale, have the ability, anti-tumor ability, effective anti-tumor ability capable of increasing the lives of these treated animals, even after a long time. At the time of euthanasia, these animals, the ones with inoculated with KT cells, they're still present in the bodies of these animals. So the idea of this project, obviously, is to make sure that our genetic modification tool and the manufacture of KT cells, to make it viable, to make it feasible, and now increase the scale of production of those cells. So the production flow for those cells is through peripheral blood from patients. We do electrophoresis of those blood cells, and we use those bottles, 1 liter bottles called Rexim bottles, in which we're able to produce a large amount of cells. So the first outcomes, large-scale outcomes, have shown us that we have an important efficiency rate here at transmission of those cells, in which over 50% of those cells in this experiment were positive to express the chimeric receptor. Sorry, this is working on its own. It's not me. It's the gizmo I'm holding. So for us, we were able to achieve an important number of T cells at the end of the process of expansion. And these T cells have a total number of T cells carrying our chimeric receptor. And then by using the PDX model, we're also able to inoculate those animals with a primary tumor derived from patients that had leukemia, for example, acute lymphoblastic leukemia at the National Cancer Institute. And these immunodeficient animals were able to develop the tumor. And upon treating those animals with the CAR-T cells, we were able to see through the flow scintigraphy rate, we have the positive CD cells, and there was a reduction in the frequency of those cells in those animals. And finally, we measured the survival rate of those animals treated with CAR-T cells. In this model, cells derived from tumors in patients, and there's been a significant survival rate significant survival of these animals that were treated with the CAR-T cells generated according to the large-scale protocol. As a prospect for our work, we were able to generate these CAR-T cells with an important capacity, anti-tumor ability, in an amount of cells that is sufficient so that we can treat patients at the site where these patients will be catered to, will be cared for, which is a cell processing unit located at the National Cancer Institute of Brazil at low costs compared to the CAR-T cells generated through viral vectors. So I'd like to thank our group members for the assistance and the foment organizations. Thank you so much. The financing agencies. Thank you so much to all of them.